Usually when you take a walk in the woods, there's this expectation of tranquility.
Right.
You know, you picture this pastoral painting. You see a decaying log, maybe some soft green moss, a little white mushroom popping up through the leaf litter.
Yeah, it all feels incredibly still.
Exactly. We tend to view the forest floor as like basically just the static furniture of the natural world.
We really do treat it like this serene backdrop for our hikes. But I mean, the moment you step into the actual microscopic uh, biochemical reality of that exact same forest floor.
That peaceful painting just gets completely shredded, doesn't it?
No, absolutely shredded.
Yeah.
We are looking at an ecological landscape that functions as a ruthless, highly active battlefield.
Yeah.
What looks like a delicate little feature of the landscape is actually, an organism running complex biological algorithms. It's engaging in active predation, synthesizing like highly targeted chemical weapons.
And you've provided us with a stack of sources today that really just tear the veil off that peaceful illusion.
I tried to bring a good mix, yeah.
It's an amazing stack. For those of you listening, we're pulling from dense academic texts like The Diversity of Macrofungi by Dr. Nafisa Begum.
Heavy reading, but worth it.
For sure. And we're digging into the highly specific Claudius Project, which catalogs poisonous mushrooms in Virginia.
Right.
And then cross-referencing all of that with practical field guides like common Kansas mushrooms. But to ground the science in real-world human experience, which I always love.
Oh, the forums.
Yes. We also have dispatches from grassroots foraging forums. Specifically, there's this wildly entertaining thread from Canadian gun nuts and also the Ecology community on Reddit.
Yeah, this eclectic mix of materials, it all points toward one specific organism that challenges, well, almost everything we assume about how a simple fungus is supposed to behave.
So our mission today for this deep dive is to explore the shape-shifting, bacteria-hunting, and, frankly, biochemically treacherous world of Coprinopsis variegata.
Commonly known as the Scaly Ink Cap.
Right. And just to set the baseline for how strange this gets for you guys listening, this is a mushroom that hunts and eats living soil bacteria.
Which is wild.
It actively dissolves its own body into a puddle of black goo. And it acts as a highly specific chemical booby trap if you happen to drink a glass of wine with your dinner.
It's quite the resume.
Okay, let's unpack this.
Well, to understand the sheer strangeness of this organism, we really have to start with its physical presence in the world.
Okay.
Before we even touch on its complex chemistry or, its predatory habits, we need to map out how rapidly and dramatically this mushroom changes its physical appearance.
It's a masterclass in morphological transformation.
Exactly.
So let's set the visual stage. Imagine you're walking through the woods in early summer or fall, looking at a decaying hardwood log.
Right.
According to the Common Kansas Mushrooms Guide and the mushroom expert source, when Coprinopsis variegata first emerges, it doesn't look anything like a classic mushroom.
No, not at all. You won't see a stalk and an open umbrella cap.
Right. It starts out as this small, maybe 1 to 3 centimeter oval or bell shape. But the most striking feature is that it is completely encased in what mycologists call a universal veil.
And the universal veil is a crucial identification feature. Understanding its mechanics is just fascinating.
What exactly is it?
Well, the sources describe it as this felty, whitish, or yellowish membrane. Think of it structurally like a soft, fibrous eggshell.
Oh, an eggshell. Okay.
Yeah, and it completely surrounds the embryonic fruiting body of the mushroom. The Claudius Project actually describes this mechanical concept beautifully when discussing a different genus, Amanita.
What do they say?
They note that as the mushroom grows, this veil resembles an egg that physically splits open as the expanding cap pushes through from the inside out.
Wow.
And the same hydraulic principle applies to the Scaly Ink Cap.
Let's pause on that word for a second. Hydraulic. Because a mushroom isn't growing the way an animal grows, right? It's not slowly accumulating bone and muscle over years.
No. Fungal growth is largely driven by water. The cells in the primordial mushroom, they're already formed. They're just miniature.
Oh, I see.
So when the conditions are right, the underlying mycelium pumps massive amounts of water into those cells.
Like blowing up a balloon.
Precisely like microscopic water balloons. This generates immense turgor pressure, hydraulic pressure basically, that forces the mushroom up out of the wood.
And that pressure is what destroys the veil.
Exactly.
Because that little felty, whitish yellow pod just can't stretch enough to contain the expanding cap. And the sources say the cap can reach up to, 7.5 centimeters across?
Yeah, that's right. So the veil literally tears apart.
And it leaves these distinctive, large, loose, felty patches all over the surface of the cap.
And beneath those torn patches, the actual surface of the expanding cap is revealed to be this grayish or brownish yellow color.
So you end up with this pale, torn fuzz sitting on top of a darker, smoother background.
Yeah, it gives the mushroom this rugged, almost woolly appearance.
Which is the direct inspiration for its common name, the Scaly Ink Cap.
You got it. But the cap expanding is really just the first act.
Right, because the gills have their own whole thing going on.
Oh, they go on a wild chromatic journey of their own.
Those are the thin, papery, spore-producing ridges underneath the cap, for those wondering.
Yes. So when the mushroom is young, the gills are pure white.
Okay.
But as the spores begin to mature, the gills start turning grayish, they deepen to a purple gray, and finally, they turn a deep, opaque, jet black.
Jet black, and that black color is the harbinger of arguably the most insane part of this mushroom's life cycle.
What's fascinating here is that the blackening of the gills isn't merely a pigment change. It's not just a color shift.
What is it then?
It is the visual evidence of a biological process called deliquescence.
Deliquescence.
Yes, this is the defining characteristic of the coprinoid mushrooms, the ink caps.
Okay.
The Diversity of Macrofungi text and the Claudius Project go into great detail about this mechanism. The mushroom does not simply dry up and blow away.
Like a normal mushroom might.
Right. Nor does it passively rot from bacterial decay. It initiates a highly coordinated sequence of self-digestion.
It melts itself. I mean, I was looking at the Reddit thread from Ecology that you included in the sources.
The one posted by Gran Trevino.
Yes.
Huh.
Gran Trevino. They documented this process in real time, showing a 5-day progression.
It's incredible to see.
Over a span of just a few days, the solid gills and the flesh of the cap actively dissolve into this thick, black, inky sludge.
Yeah.
The cap edges become tattered, they curl upward, and you get these hanging globs of black spores and like partially digested mushroom flesh just dripping onto the forest floor.
And the chemistry behind this is an incredible feat of biological engineering.
It has to be.
I mean, this is an active, internally programmed sequence driven by autodigestive enzymes called chitinases.
Chitinases.
Right. We have to look at the cellular structure of fungi to understand why this matters. Because the cell walls of fungi are not made of cellulose like plants are.
They're made of chitin, which the Kansas Field Guide makes a really interesting point about that. They note that fungi are actually more closely related to lobsters and insects than they are to plants.
Which blows people's minds, but it's true. Because chitin is the exact same tough, complex carbohydrate polymer that makes up the exoskeletons of arthropods.
So a lobster shell and a mushroom wall are made of the same stuff.
Exactly. It is incredibly strong and rigid.
Okay.
So to break that structure down, the mushroom must synthesize these chitinases. There's specialized enzymes that act like molecular scissors.
Molecular scissors.
Yeah, they seek out the specific chemical bonds holding the chitin polymer together, the beta-1-phthalate glycosidic bonds, to be technical and hydrolyze them.
They just snip them apart.
They sever the connections. The mushroom is literally manufacturing the specific chemical tool needed to dismantle its own structural scaffolding from the inside out.
But wait, why? I mean, why would an organism spend precious energy gathering nutrients, pumping water to build this complex, fleshy, fruiting body with a stem and a cap and thousands of delicate gills just to initiate a chemical self-destruct sequence a few days later?
It seems counterintuitive, I know.
It feels like spending months building a beautiful, intricate wooden house, and the moment you finish the roof, you just deliberately set the foundation on fire. What is the evolutionary logic there?
Well, evolutionary biology evaluates success purely through the lens of reproduction.
Right, sure.
To understand the why, we have to contrast coprinoid mushrooms with typical gilled mushrooms.
Like a portobello.
Exactly. A standard mushroom, a portobello, or a white button mushroom relies entirely on wind to disperse its spores.
Right. The spores drop and blow away.
The gills stay dry, the mature spores detach, and the ambient breeze carries them off. But the Scaly Ink Cap has abandoned that strategy entirely.
Because the spores are trapped in that wet mucus liquid it just created, they physically cannot blow away.
They are glued into the sludge.
So how do they spread?
The diversity of macrofungi source explains this brilliance by drawing a parallel to a closely related ecological strategy found in stinkhorn fungi.
Oh, stinkhorns! Those are the ones that smell terrible.
Very terrible, yes. Because the spores are suspended in a thick, gelatinous mucus matrix, wind dispersal is completely impossible. Instead, the liquid itself is the delivery mechanism.
How does that work?
It is highly attractive to insects.
Wow.
Flies and beetles arrive to feed on the mucilaginous sludge, or they simply walk through it while exploring the decaying log.
Oh, that makes so much sense.
Yeah, the sticky spore ink adheres to their feet and bodies. When the insects fly off, they carry the spores to completely new, distant locations.
So it's basically outsourcing its reproduction to local insect traffic. The text actually compares this directly to how flowers use nectar to attract bees for pollination.
It's an exact parallel. The melting isn't a failure of the organism. It is a highly evolved, specialized dispersal mechanism. It's designed to utilize animal vectors rather than unpredictable wind currents.
And that biological ink is so effective that humans have figured out how to use it too.
Oh, yeah.
The Claudius Project mentions this in their fun fact section, and that Reddit user we mentioned, Gran Trevino, was actively experimenting with it. You can literally use the black sludge dripping off this mushroom as drawing or painting ink.
It's amazing. The high concentration of dark spores suspended in the autodigested liquid creates a surprisingly viable natural pigment.
How do you even collect it?
In amateur mycology circles, foragers will collect the deliquescent caps in a glass jar. They let the enzymatic process finish completely.
So it's just a jar of black liquid.
Pretty much. And then perhaps add a drop of clove oil to act as an antibacterial agent, just to prevent mold.
Oh, smart.
And then you can dip a calligraphy pen straight into the jar and write with it. It's a wonderful synthesis of art and biology.
And there was another Reddit user in that same thread, Stace Untamed, who asked about preserving the whole life cycle in resin.
Yeah, I saw.
It just demonstrates just how visually arresting this whole transformation is for people.
Truly.
Yeah.
But, if the mushroom is capable of breaking down its own complex cellular structure with that much biochemical precision,
Right.
we have to ask what it's using to build itself up in the first place.
Before it melts the house, how is it gathering the lumber?
Exactly. Because the ultimate mushroom source reveals it is doing far more than passively drinking water from a dead log.
So to map its nutritional strategy, we first have to establish its baseline ecological role. Coprinopsis variegata is primarily classified as a saprobe.
Yes.
Let's define that really quickly using the glossary from the Kansas Guide. Saprobic means an organism uses dead or decaying organic material as food, basically aiding in decomposition.
Right. And the diversity of macrofungi text emphasizes how vital saprotrophic macrofungi are to the survival of terrestrial ecosystems.
They're the cleanup crew.
They are the primary engines of organic decomposition in forests. Plant matter, especially the wood of dead trees, is packed with highly recalcitrant, stubborn polymers like lignin. Lignin is essentially a random, incredibly complex web of carbon rings that most organisms, including almost all bacteria, just cannot break apart.
It's nature's plastic. It is incredibly durable.
To breach that armor, saprobic fungi exude lignin-modifying enzymes into the wood.
Oh, so they just melt the wood too.
In a way, yes. Specifically, they use peroxidases and lactases. These are brute force oxidative enzymes. They blast apart the complex chemical bonds in the lignin, exposing the softer cellulose underneath.
And then they eat the cellulose.
Exactly. The fungus breaks that down into simple sugars for energy. By dismantling this dead wood, the fungus releases carbon that was fixed during photosynthesis back into the soil, along with essential minerals.
So without saprobes acting as the demolition crew, the forest would essentially just choke on a pile of its own undecomposed dead material.
We'd be buried in dead trees, yeah.
So on a baseline level, the Scaly Ink Cap is doing that vital cleanup work. The mushroom expert source notes it's commonly found growing in large, loose clusters on well-decayed hardwood logs or on wood chips.
Right, sometimes you see them popping up in the middle of a lawn.
Yeah, but that's just because the wood they are digesting is buried underground.
Exactly.
So that is the standard saprobic bay job. But the ultimate mushroom profile drops an absolute bombshell detail about a secondary feeding behavior.
This is where it gets crazy.
Coprinopsis variegata isn't just a wood rotter. It is a bacterial predator.
A predator.
It actively hunts and digests living soil bacteria, specifically targeting genera like Pseudomonas and Agrobacterium.
When we look at soil ecology, this behavior just completely upends the traditional hierarchy of the food web.
How so?
Well, we are taught that bacteria are the ultimate microscopic decomposers that eventually consume everything else, right? Including dead fungal matter.
Yeah, that's what you learn in high school biology.
But here, the fungus has evolved a mechanism to hunt the bacteria.
But how does a stationary organism with no eyes, no nervous system, and no muscles hunt evasive prey?
It relies on a mechanism called a chemoattractive response.
Oh, okay, let's break that down.
To visualize this, we have to look beneath the soil at the mycelium. The mycelium is the vast hidden network of microscopic root-like threads called hyphae.
Right.
These hyphae grow exclusively at their tips, pushing through the soil and wood, and they are highly sensitive to chemical gradients in their environment.
So they're essentially tasting the dirt as they push through it.
That's a great way to think about it. When a bacterial colony like Pseudomonas is metabolizing, it excretes waste products and specific chemical signatures into the surrounding moisture.
And the fungus can taste that.
Yes. The receptors on the growing tip of the fungal hypha detect those molecules. If the concentration of the bacterial chemical is stronger on the left side of the hypha than on the right, the fungus alters its internal cellular scaffolding.
It changes shape.
It directs secretory vesicles, little packets of growth material, to fuse with the cell membrane disproportionately on the side facing the chemical gradient.
So it physically bends its growth trajectory toward the source of the chemical trail.
Exactly.
It's like a microscopic heat-seeking missile, but tracking a chemical exhaust signature instead of a thermal one.
It steers directly into the bacterial colony.
And then what happens?
Once the hyphae make physical contact, they shift their chemical output. They stop exuding wood-rotting enzymes and begin secreting targeted, specialized digestive enzymes that dissolve the cell walls of the living bacteria.
Wow.
The fungus that absorbs the liquefied cellular contents. It is a ruthless and shockingly efficient mechanism. The source notes the mycelium can detect, engage, and consume entire bacterial colonies in less than 24 hours.
But why go through the trouble of evolving a complex chemical tracking system to hunt bacteria when you're already living inside a gigantic all-you-can-eat buffet of a dead log?
It comes down to nutritional bottlenecks.
Nutritional bottlenecks.
Wood is incredibly rich in carbon, which provides energy, but it is notoriously deficient in nitrogen.
Oh, I see.
And nitrogen is the fundamental building block for proteins, DNA, and the chitin needed to build the mushroom itself.
So it's starving for nitrogen.
Exactly. Bacteria, on the other hand, are highly concentrated little packets of nitrogen and other essential micronutrients.
They're like little vitamin pills.
Yes. By evolving the ability to predate on bacteria, the Scaly Ink Cap bypasses the nitrogen bottleneck of its woody environment. This gives it a massive competitive advantage over other fungi that strictly rely on decaying plant matter.
So if we recap for a second, we have an organism that starts as a white egg, bursts into a scaly cap, melts itself into a puddle of black ink, and spends its time underground running chemical targeting systems to dissolve living bacteria.
That sums it up.
Because its morphology is so fluid and its true nature is hidden underground, it has caused an unbelievable amount of confusion for human scientists trying to classify it.
Taxonomy, the science of naming, describing, and classifying organisms, is notoriously fragile when it comes to fungi.
Yeah.
And Coprinopsis variegata is a textbook case study in the limitations of early mycology. The historical record for this specific mushroom is just a tangled mess of overlapping names and chronic misidentifications.
The mushroom expert source details this incredible historical struggle centered around a guy named C.H. Peck.
Oh, Peck.
Yeah, Charles Horton Peck. He was a giant in the field, a pioneer in American mycology working in the late 1800s.
Very prolific.
But over a period of 24 years, from 1873 to 1897, Peck basically kept discovering this exact same mushroom out in the woods over and over again, and he gave it new names every time.
Right.
He described it 3 separate times, publishing it as Coprinus variegatus, then later as Coprinus bulbosus, and finally as Coprinus quadrifatus. How does a leading expert of that era make that kind of seemingly obvious mistake 3 times in a row?
Well, we have to forgive Peck because he was working exclusively with morphological features.
Meaning just what it looked like.
Right, what he could see with his naked eye and a basic microscope. Fungi do not have the rigid, genetically locked developmental blueprints that mammals do.
Okay.
A golden retriever will generally always look like a golden retriever regardless of where it grows up. But a mushroom's physical appearance is highly plastic. It's heavily dictated by immediate environmental conditions, like ambient moisture, temperature fluctuations, and the specific density of the wood it's consuming.
So let's look at the specific features that threw Peck off the scent. He named one of his iterations bulbosus. Why?
Because the stem of this mushroom frequently terminates in a large, pronounced, rimmed bulb that acts as an anchor deeply inserted into the wood.
Oh, a bulb, hence a bulbosus.
Exactly.
That bulb isn't always there. If a specimen happens to grow in a tighter crevice without developing that pronounced bulb, or if pecks simply snap the stem off above the woodline without realizing the bulb was left behind in the dirt, the specimen suddenly lacks a key diagnostic feature.
Right.
So it looks like a completely different species to him.
And the veil patches are another massive variable.
Yeah.
We discussed how the universal veil tears into those felty scales.
Yeah.
The Kansas Guide specifically advises foragers to always consider recent moisture amounts when identifying. Heavy rain can act as a physical abrasive.
Oh, so it washes the scales off?
Yes. If a severe thunderstorm washes those loose patches off the cap, the mushroom goes from looking woolly and scaly to completely smooth and bald.
Wow.
So if you are a 19th century botanist and you have a bald mushroom with no bulb sitting on your desk next to a scaly mushroom with a bulb, the logical scientific conclusion based on morphology is that you are looking at two distinct species.
That makes total sense. It wasn't until 1979 that a researcher named Patrick published a comprehensive analysis looking at all these variations and concluded they were just physical iterations of the exact same organism.
He synonymized them.
Synonymize them. And under the rules of botanical nomenclature, the oldest validly published name takes priority.
Which was Peck's 1873 name, variegatus.
And then later on, broader genetic studies shifted the entire genus from Coprinus to Coprinopsis, giving us the modern name we use today, Coprinopsis variegata.
That 1979 synonymization brought a sense of order to the chaos. But modern science has recently introduced a massive plot twist that threatens to unravel the taxonomy all over again.
I love a good taxonomic plot twist. The mushroom expert source points out that modern DNA research, specifically a 2020 study by a team led by Douglas, utilized a single gene alignment and found considerable phylogenetic separation between old collections labeled Quadrifatus and Variegatus in GenBank.
Yes.
So the DNA is suggesting that Peck might have actually been right the first time, and they might be entirely different species.
This is a perfect window into the current, often messy transition phase of modern mycology. We are caught between a historical system based entirely on what a mushroom looks like and a modern system based on its underlying genetic code.
Let's pause on single gene alignment. What does that actually mean when a scientist does that in a lab, and why is it causing this controversy?
Well, when scientists want to genetically identify a fungus, they don't usually sequence the entire genome because that is incredibly expensive and time-consuming.
So what do they do?
Instead, they look for specific short sections of DNA that act like a barcode. In mycology, the standard barcode is the ITS region, the internal transcribed spacer.
The ITS region.
Yes. This is a piece of non-coding DNA situated between the genes that produce ribosomal RNA. Because it doesn't code for vital proteins, it isn't subject to the same intense evolutionary pressure.
Meaning it mutates faster.
Exactly.
Yeah.
It mutates at a faster, more predictable rate. So by extracting DNA from a mushroom, using PCR to amplify that specific ITS region, and comparing the sequence of base pairs to a global database like GenBank, scientists can see how genetically distant two specimens are.
Okay, so the Douglas study looked at the ITS barcodes for these older specimens and found they didn't match closely enough to be the same species. There is phylogenetic separation.
Correct.
But if the DNA says they are different, why do modern field guides and sources still lump them all together under one name, Coprinopsis variegata? Why ignore the DNA?
Because the scientific method requires exhaustive proof before overturning established taxonomy, and biology is inherently conservative.
So a single gene isn't enough proof.
A single gene alignment is a powerful clue. It's a preliminary red flag that there is hidden, cryptic diversity here. But a single gene can be misleading due to phenomena like incomplete lineage sorting, where different genes within a population evolve at different rates.
I see.
To officially split these mushrooms back into separate recognized species, mycologists need to conduct multi-gene sequencing to confirm the divergence across multiple parts of the genome.
And there's an incredible historical hurdle to doing that, right? You can't just sequence any random mushroom you find in the woods and apply Peck's old names to it.
Precisely the problem. To officially tether a genetic sequence to the historical name Quadrifatus or ebulbosus, scientists have to sequence the DNA directly from Peck's original type collections.
His original mushrooms from the 1800s.
Yes. A type collection is the literal physical, dried mushroom specimen that Peck collected back then, which is carefully preserved in a university herbarium somewhere.
That sounds different.
They have to extract viable DNA from a century-old dried husk, sequence it, and figure out which modern genetic profile belongs to which historical name.
Wow.
Until that painstaking, highly technical work is completed and published, the mycological community maintains a holding pattern, keeping them lumped under the oldest valid name, Coprinopsis variegata.
This academic confusion over identity isn't just locked away in university herbariums, though. It trickles right down to the everyday person walking their dog or trying to forage meal in the woods.
Oh, absolutely.
If the world's top mycologists armed with microscopes and DNA sequencers are confused, amateur foragers are absolutely struggling. We see this perfectly illustrated in that Canadian gun nuts forum thread included in our sources.
That forum thread is just a brilliant, unvarnished look at real-world identification struggles in the field.
So, a user named Mount Sweetness posts pictures of a mushroom growing out of a lawn and asks the community for an identification. They make an educated guess that it might be a scaly ink cap, which they refer to as Coprinus quadrifidus, interestingly using one of Peck's older synonymized names.
That is interesting.
But then another user, Smiley1, jumps into the thread and insists that some of the pictures are actually shaggy manes.
The classic mix-up.
Mount Sweetness pushes back immediately, arguing that shaggy manes are much larger, typically 5 to 15 centimeters across, while their specimens are only 2 to 8 centimeters. And they point out that they are all growing from the same cluster.
That back-and-forth debate highlights the true lookalikes that foragers really have to navigate. The shaggy mane, Coprinus comatus, is a classic, highly sought-after edible mushroom.
A good one to eat.
Yes, and while it is closely related and also undergoes that dramatic deliquescence into black ink, it possesses a distinctly different physical architecture.
What are the key features that separate them in the field for anyone listening who wants to forage?
Well, the cap of the shaggy mane is much more cylindrical. It's almost shaped like a tall, narrow bullet or a closed umbrella.
Okay, tall and narrow.
And the scales on a shaggy mane are also structurally different. They are deeply shaggy, lifting away from the cap like peeling shingles on a roof.
Like peeling shingles. Got it.
Whereas the Scaly Ink Cap has those looser, felty patches left over from the universal veil tearing.
The ultimate mushroom source points out two other very close relatives that further muddy the waters. There is Coprinopsis atramentaria, which is incredibly similar in size, color, and habitat, but it completely lacks those patchy, woolly scales.
Right, it's smooth.
And then there's a species that sounds like an absolute nightmare to differentiate. Coprinopsis straussmayeri.
Yes, straussmayeri is the true taxonomic headache for a field forager because the morphological overlap is severe.
How do you even tell them apart?
However, there are crucial ecological and structural differences if you know exactly what to look for. As mushroom expert notes, the Scaly Ink Cap is fundamentally a woodland species growing on well-decayed logs.
Okay, woodland.
Straussmayeri, on the other hand, is an urban opportunist.
Meaning you find it in the city?
It thrives in wood chips, on the stumps of recently cut street trees, or following the dying root systems of those cut trees beneath the soil, which gives it the illusion of growing directly out of the dirt in suburban environments.
But beyond location, how do you tell them apart visually?
Physically, the universal veil on straussmayeri breaks into much finer, smaller scales rather than large patches.
Okay, finer scales.
It usually lacks that pronounced bulbous base on the stem that Peck observed. And most distinctively, straussmayeri features reddish-brown rhizomorphs at the base of the stem.
Let's explain rhizomorphs, because that's a fascinating fungal adaptation. They look like roots, but fungi don't have roots.
Right. Rhizomorphs are essentially thick, tough cords created when thousands of individual microscopic hyphae aggregate and bundle together, forming a hardened outer rind.
What do they do?
They function like biological pipelines. They allow the fungus to transport water and nutrients over significant distances through the soil, connecting different food sources.
So finding those reddish-brown cords at the base of a scaly, melting mushroom is a strong indicator of straussmayeri.
Exactly.
So we have established a mushroom that melts itself to spread spores, uses chemical tracking to eat bacteria, and looks like 3 different species depending on the weather, the DNA, or the decade.
It's a lot.
But misidentifying this mushroom isn't just an academic problem or fodder for a forum argument. It can lead to a highly unpleasant systemic physiological crisis if you decide to take it into your kitchen.
This is the real danger.
This brings us to the Coprine conundrum, which is essentially a biological chemical booby trap.
This is where the academic details intersect with visceral, dangerous consequences for human foragers. The edibility of the scaly ink cap is a subject of intense, heavily debated scrutiny.
Let's look at the wildly conflicting advice available to the public. The Eat the Planet source outlines this perfectly. It notes that the scaly ink cap's edibility is heavily debated.
Very debated.
Some foragers passionately defend it, considering it to be the tastiest of all the ink caps, praising its texture and flavor profile. Yet other sources completely dismiss it, describing the taste as terribly bitter and of poor culinary value.
Taste is subjective, but the older field guides are what worry me.
Right. If you consult older, traditional field guides, you will frequently find it listed under the ominous category of edible with caution.
Edible with caution is a phrase that should trigger an immediate red flag for any responsible forager.
Definitely.
The reason for that caution is fascinating because it differs entirely from how most toxic mushrooms operate.
How so?
Well, the scaly ink cap is not inherently deadly like an Amanita aethaloides, the death cap.
The death cap will just straight up kill you.
Right. A death cap contains amatoxins, which will irreversibly halt protein synthesis in your liver and kidneys and kill you, regardless of what else you consume or do.
Okay.
The danger lurking within the scaly ink cap is highly conditional. It harbors a specialized biochemical compound called Coprine.
And here is the absolute wildest part of this entire deep dive. Coprine itself is fundamentally harmless.
Completely harmless on its own.
You could theoretically go out into the woods, accurately identify a fresh scaly ink cap, take it home, saute it in butter, eat a plate of it, and feel absolutely fine. Your digestive system would process the mushroom, you'd absorb the nutrients, and you would walk away completely unscathed.
However, if you introduce alcohol into your system, the entire physiological landscape of your body is hijacked.
Hijacked!
The Claudius Project document details exactly how this trap is sprung. Coprine intercepts and paralyzes a highly specific metabolic pathway in the human liver, specifically targeting an enzyme called aldehyde dehydrogenase.
To understand the sheer brutality of this trap, we need to unpack the chemistry of human alcohol metabolism.
Okay.
When you drink a glass of wine or a beer, your body recognizes ethanol as a toxin and initiates a two-step process to eliminate it. Step 1. An enzyme called alcohol dehydrogenase breaks the ethanol down into a highly toxic, highly reactive compound called acetaldehyde. Acetaldehyde is the chemical primarily responsible for the terrible symptoms of a severe hangover.
And that is the crucial intermediate stage. Normally, your liver immediately transitions to step 2. It deploys the second enzyme, aldehyde dehydrogenase, to rapidly break that toxic acetaldehyde down into acetic acid, which is essentially just vinegar.
And vinegar is harmless.
Your body then easily flushes the harmless acetic acid out of your system.
But Coprine throws a wrench into that machinery. How exactly does it block that second step?
It's a brilliant piece of biochemical sabotage. When you consume the mushroom, the Coprine is metabolized in your body into an active compound called 1-aminocyclopropanol.
That's a mouthful.
It is. But this new molecule travels to the liver and physically enters the active site of the aldehyde dehydrogenase enzyme. It binds to it, blocking the site completely.
So the enzyme is broken.
The enzyme is paralyzed. It cannot process the acetaldehyde.
So you've had a glass of wine, your body has converted it into toxic acetaldehyde, but the exit door is locked.
Yeah.
What happens to the human body when that highly toxic backlog of acetaldehyde floods the bloodstream?
The resulting physiological crisis is clinically known as a desulfuram-like effect, or an antabuse reaction.
Antabuse.
Yes, antabuse is the commercial brand name for the drug desulfuram, which was historically prescribed by doctors to treat chronic alcoholism.
I've heard of that.
The entire pharmacological premise of the drug was to force sobriety by making the physical act of drinking alcohol a punishingly awful, terrifying experience. Coprine induces the exact same violent reaction, but naturally.
And the symptoms detailed in the Claudius Project are harrowing.
Yeah.
If you eat this mushroom and then consume alcohol, you will experience intense, burning facial flushing. You get severe nausea, followed by violent, uncontrollable vomiting. You develop a splitting, throbbing headache, a rapid and irregular heartbeat tachycardia, dangerously low blood pressure, a prickling or tingling sensation in your extremities. And in severe cases, it can escalate to altered mental status, confusion, and even seizures. Your body physically rebels against the poison circulating in your veins.
As severe as the immediate symptoms are, the most insidious aspect of this interaction, the true trap, is the time delay.
This is the part that blows my mind, because the danger isn't just about drinking a glass of wine while you eat your mushroom dinner. The metabolized Coprine compound binds to those liver enzymes so effectively that it stays active in your system, keeping that pathway shut down for up to 5 days.
It is irreversible enzyme inhibition. Your body physically has to manufacture entirely new enzymes from scratch to replace the paralyzed ones, and that process takes days.
So you could eat a Scaly Ink Cap on a Monday afternoon for lunch, experience absolutely zero symptoms, digest it, and forget all about it. Then you go out for a celebratory beer on Thursday night, 3 days later, and suddenly find yourself violently ill, your heart racing, vomiting in an alley. You would likely assume you had severe food poisoning or a sudden, massive allergic reaction. You would never mentally connect the terrifying medical episode on Thursday to the completely benign-feeling mushroom you ate on Monday.
It is a remarkable piece of delayed action chemistry that makes foraging for this genus a high-stakes gamble.
Absolutely.
Because of the severity of this reaction and new emerging science, the modern consensus on the edibility of the Scaly Ink Cap has shifted dramatically away from those older field guides.
Right. The Claudius Project notes that the North American Ecological Association, known as NEMA, issues a very strict, uncompromising warning regarding Coprine.
What is their stance?
Beyond the immediate violent reaction to alcohol, recent toxicological studies have demonstrated that Coprine is actually carcinogenic. It is cancer-causing in mammalian models.
Oh, wow.
Because of the dual threat of the antabuse reaction and the long-term cancer risk, NEMA advises against eating Coprinopsis variegata or any coprine-containing mushroom entirely. It is simply not worth the biological risk.
It provides a fascinating contrast and risk assessment when you look back at the anecdotal stories of foragers. Returning to that Canadian Gun Nuts forum, there is a user named Kimluki who tells a wild, vivid story from his youth that perfectly illustrates how thin the margin of error is.
Oh, I love this story.
He and a buddy were out hunting grouse near Spence's Bridge in British Columbia. He decides to cook up a massive frying pan full of shaggy manes right alongside their grouse and washes it all down with a bottle of cheap, potent alcohol.
His city slicker friend is watching this happen, absolutely terrified. He's fully expecting Tim Lookie to drop dead right there in the bush because he knows the legends about ink caps and alcohol.
But Kamluki was completely fine. He suffered no ill effects because he was eating shaggy manes, Coprinus comatus.
And shaggy manes don't have it.
Exactly. Despite being closely related to the Scaly Ink Cap and also melting into black ink, shaggy manes do not synthesize the Coprine compound.
That is so lucky.
The story ends hilariously with the terrified buddy eventually joining in. And on the drive home, every time he spots a white mushroom head popping out of the dirt, he yells at Kamluki to stop this truck so he can run out and harvest more.
It's a great story of a successful, albeit reckless-seeming foraging trip, but it perfectly highlights the razor-thin margin of error in amateur mycology.
Yes, it does.
Kamluki was safe because he accurately identified his target. But if someone read that forum post, felt inspired, marched into the woods, and mistakenly harvested the Scaly Ink Cap instead of the shaggy mane, perhaps because heavy rains had washed the scales off and then cracked open a beer around the campfire,
Oh boy.
that hunting trip would have ended with a frantic, terrifying rush to the nearest emergency room.
The fungal kingdom does not tolerate casual assumptions. It demands intense respect, precision, and a deep understanding of the underlying biochemistry. The Scaly Ink Cap is not a casual woodland encounter. It is a highly specialized, formidable organism.
So let's pull back and summarize the incredible journey of this single species we've explored today.
Let's do it.
We started with this delicate, white, fuzzy, shape-shifting fruiting body that looked like it belongs in a children's fairy tale, expanding until it tears its own veil. We learned that it orchestrates its own self-destruction, synthesizing specific enzymes to melt its chitin scaffolding down into a puddle of black ink that human artists used to draw with, relying on flies to carry its spores away.
Such a wild life cycle.
Then we look beneath the soil and realize it is a microscopic predator, utilizing complex chemical tracking systems to actively hunt down and dissolve living bacterial colonies to steal their nitrogen. We saw how its extreme physical variability has caused over a century of taxonomic headaches, forcing modern scientists to sequence ancient DNA just to figure out what to call it.
It's a taxonomic nightmare.
And finally, we uncovered the delayed-action chemical booby trap it hides within its flesh, lying in wait to severely punish any foraging mammal that dares to pair it with alcohol.
It is a stunning, almost unbelievable portfolio of biological adaptations and chemical warfare strategies packed into a mushroom that rarely grows larger than 3 inches across.
For you, our listener, I think this deep dive proves something profoundly exciting. You do not need to look to science fiction novels or theorize about distant alien planets to find mind-bending, bizarre biology.
Not at all.
There is an intense, invisible war of chemical attractants, autodigestive enzymes, hydraulic growth, and systemic delayed-action toxins happening right under our feet, perhaps even buried in the damp wood chips of your own backyard. The natural world is endlessly, aggressively fascinating once you know how to look at it.
I want to leave you with one final thought to mull over regarding the scaly ink cap, specifically looking at that chemical trap. We know that this fungus produces Coprine, a highly specific chemical compound that acts as a prodrug, perfectly mimicking human liver substrates to paralyze aldehyde dehydrogenase and cause severe, unforgettable physical suffering when combined with fermented fruits or grains.
Right.
Why does this mechanism exist? Is this purely a bizarre, random chemical coincidence? Or, when we consider how rigorously evolution shapes organisms for survival, is this a deliberately evolved defense mechanism?
That's a great question.
If the mushroom's entire reproductive strategy relies on melting into a puddle of ink so insects can carry its spores, a foraging mammal coming along and eating the cap before it melts completely destroys that reproductive cycle. Fungi do not have teeth, claws, or legs to run away, so they fight with complex chemistry. Is Coprine an evolutionary weapon designed specifically to teach foraging mammals a delayed, painful lesson about what not to eat? The true depth of fungal chemical warfare and the hidden ecological intent driving it is something we are only just beginning to decode.
The next time you take a walk in the woods and you see a peaceful little mushroom resting quietly on a decaying log, remember that static backdrop is an illusion. You are walking through an active biochemical battlefield. Thanks for diving deep with us today.